There are several pitfalls to avoid when staining prior to immunodetection. Before you start, make sure that the stain is compatible with your choice of membrane. Secondly, you must be certain that the stain does not interfere with antigen-antibody binding. And if the stain is reversible, you need to be certain that the chemistry does not affect antigen epitopes in a detrimental way. When considering pre-labeling strategies, such as the Bio-Rad stain-free technology and the Amersham Western blot system, there are several points to keep in mind. Both methods couple fluorescent dyes to sample proteins for visualization. The stain-free technology uses a trihalo compound in the gel matrix that is coupled to proteins by UV cross-linking. The Amersham system uses an amine-reactive Cy5 dye for sample labeling prior to electrophoresis. These methods are sensitive to biological variability because labeling is directed against a specific amino acid. In the stain-free system, it's tryptophan. The Amersham Cy5 system targets primary amine groups on lysine residues. Because amino acid composition is variable, protein-to-protein variation is expected, and the extent of labeling can be inconsistent across your samples. Chemical modification of epitopes can introduce variability in a variety of ways. First, as discussed earlier, labeling can be variable due to amino acid composition of sample proteins. Secondly, it can interfere with electrophoresis or transfer. And finally, it may also inhibit antibody binding. Here is an example where Bio-Rad scientists show how stain-free labeling can affect quantitative analysis if an epitope contains a TRP residue. Before UV cross-linking on the control blot, the green line shows an increasing signal as higher amounts of hemoglobin are loaded. However, when the blot is subjected to UV cross-linking, the antibody binding is disrupted and the black line demonstrates a dramatic reduction in signal. Overall, normalization by total protein staining is a very relevant loading control that reduces the impact of biological variability, especially when applied as staining of membranes after transfer. As scientific publishers look more closely at experimental methods and data analysis, it presents an important opportunity to re-evaluate commonly used normalization methods. We have talked about the different strategies to better understand how they work, how they can fail, and how they affect reproducibility.